My name is Abdullah Sattar. I am currently a senior pursuing a major in biology. I am an undergraduate researcher in the Cellular and Molecular Brain Research Laboratory of Dr. Jefferson Kinney. Our lab is investigating the mechanisms between Alzheimer's disease and neuroinflammation. My project is entitled Examination of GABA B receptor levels after poly-IC and baclofen administration in wild-type female mice. Neuroinflammation is defined as the inflammation of the brain. In situations of trauma or infection, this response is perfectly normal. Inflammation allows immune cells to deal with any damaged cells or infectious agents. However, in Alzheimer's disease, this neuroinflammation is long-lasting, otherwise known as chronic, and leads to increased damage of the neurons and an increased formation of amyloid beta plaques as well as the hyperphosphorylation of tau. The cells responsible for immune signaling in the brain are called microglia, and there has been evidence that anti-inflammatory signaling molecules, known as cytokines, can be increased by manipulating the GABA B receptor induction. The GABA B receptor is activated by gamma-aminobutyric acid, a common neurotransmitter used in the brain. The GABA B receptor is divided into two subunits, B1 and B2. B1 is further divided into an A and a B subunit, which will be discussed later. If the GABA B receptors can be manipulated, it could pave the way for an effective treatment for Alzheimer's disease. The drug baclofen was utilized as a GABA agonist. This means it has the ability to mimic the GABA neurotransmitter. Poly-IC was used to induce inflammation. This project utilized female mice in order to provide a new viewpoint in Alzheimer's disease research. The mice were 6 months old and were divided into 4 treatment groups, namely saline-saline, saline-baclofen, poly-IC saline, and poly-IC baclofen. Before treatment administration, estrus cycles were lined up using male pheromones from soiled bedding. After 3 days and visual confirmation, the treatments were started. 2 groups were injected with saline, while the other 2 were injected with 40 mg per kilogram of poly-IC. They were injected once a day for 7 days. On the 8th day, a group of the saline mice and a group of the poly-IC mice were injected with 4 mg per kilogram of baclofen. The remaining 2 groups were injected with saline, and all the mice were sacrificed to obtain hippocampal tissues. The GABA B1 and B2 receptor concentrations were semi-quantified using Western blotting. The Western blots were analyzed using ImageLab software and SPSS. Now we go on to the results. Before we dive in, we need to understand the GABA B receptor a little better. As stated before, there are 2 parts, B1 and B2, and B1 consists of subunits B1A and B1B. B1A is mainly responsible for presynaptic, otherwise known as incoming, interactions, while B1B is responsible for postsynaptic, otherwise known as outgoing, interactions. In Figure 1A, GABA B1A receptor levels seem to be consistent throughout the treatment groups, as no significant change is observed. In Figure 1B, GABA B1B receptor expression in the poly-IC saline group are around double that of the saline-saline control group. Although the saline baclofen group has around a 40% increase in expression, it is not significantly different from the saline-saline control group. In Figure 1C, GABA B2 receptor expression from the saline baclofen group are up by around 40%, however, this is not a significant increase. Figure 2 shows a representative Western blot membrane. Overall, the only significant data was that of the GABA B1B receptor from the poly-IC saline group. Due to the postsynaptic nature of the subunit, this data shows that perhaps hippocampal cells in the poly-IC saline group are more sensitive to poly-IC. Although some groups showed promising increases compared to the control group, it wasn't enough to be considered significant. Future direction of this project is to analyze the mRNA transcripts of the GABA B1 and B2 receptors, examine the presence of inflammatory markers such as NF-kappa B, and to conduct a comparison experiment using male mice. This is the end of my presentation. Thank you for listening. I would like to thank the Office of Undergraduate Research for the financial support to make this poster possible, and to the National Institute of Health Cobre Award to Dr. Kinney.